students, now we are in chapter 3, recombinant DNA technology. So here are all of the learning outcomes. So you must know how to define DNA technology, what are the tools. Okay, within the tools, you can explain what kind of things are there and what do they do. And then you also have to be able to describe what are the cloning vectors, and then the host, and then lastly, modify enzyme and function. And after that will be the other step of cloning, okay, using plasmid as vector, which is totally different thing, PCR, and also the examples of usage of recombinants in nature, and also in other genera such as agriculture, insulin, environment. Okay, so what is recombinant DNA technology? So if you look at the name recombinants, it is from the word combine. So we can say that DNA, which are recombinants, they are a combination of more than one source of DNA. Okay, we make it into a single recombinant molecule. So this technology is based on combining two different DNAs into a DNA which is artificial. Okay, we combine DNA sequences which are not seen together in nature. Okay, so in terms of genetic modification, recombinant DNA is produced through the addition of relevant DNA into an existing organism's genome. For example, plasmid or bacteria. Okay, and then when we add that new DNA into the host DNA, they will code for a desired product like proteins. Okay, for example, proteins for immunity. So it is a little bit different from genetic recombination. Genetic recombination is actually natural. So when does it happen? It happens during the meiosis. So when the mother and father's chromosomes, okay, during the meiosis, they would shuffle their genes together in recombination. So the two resulting chromosomes will be a mix between the two. So that's actually recombination. So that's natural. That's different compared to recombinant. Recombinant is actually artificial. Human made this. Okay, so what are the tools needed for DNA or gene cloning? First of all, we must have target DNA. Okay, it is the DNA that you want to use, and then the restriction enzyme used to cut the DNA into fragments. DNA cloning vectors like plasmid or bacteriophage to carry target gene to host cell, so like a taxi. Okay, host cell, they are the one that will be the acceptor of this new DNA. And then lastly, the modified enzymes like DNA ligase and Taq polymerase. So we need this as a tool to combine DNA together, or maybe we want to clean up or whatever DNA that is left over. Okay, because we are actually not cutting DNA using scissors. We are actually using a chemical, okay, a chemical that can cut the chemical called DNA. Okay, so the first thing that is very important in cloning would be the RE, restriction enzymes. These are enzymes that can cut DNA into small pieces. So this is actually natural, okay, in certain bacteria, because certain bacteria, they use this as a defense against viruses. So viruses, they like to infect the bacteria with their own DNA, turning the bacteria into a factory for themselves. So the bacteria would develop this enzyme to cut the DNA of the virus before it infects them. And what's so special about the restriction enzyme is that it is very specific. So let's say that you have scissors, right? But the scissors can only cut green color paper or only red color paper. Very specific. It doesn't cut white or black paper. So it is something like that. Okay, restriction enzymes can only cut at certain points within the DNA. So we call it the restriction site. Okay, for example, you can see in this picture molecule A and B. So there are two different DNA molecules from two different sources. If we digest each of them with the same restriction enzyme called BAMHI, it will cut them in a way that it will form sticky ends, which are unpaired bases. And the unpaired bases will always be the same, okay, like GATCC and CCTAG. So if you can see here, the hanging ends, sticky ends, are actually paired with each other. So if you look at the red one is CTAG. Okay, if you remember, C partners with G, okay, T partners with A, and when you look at that, in molecule B, it is the partner, GATC. So they can easily be combine together with the DNA ligase to make a recombinant DNA. So transition site will be the area where it is cut. So it is 4 to 8 nucleotides in both DNA strand arranged in opposite direction, which is palindromic. So E. coli recognizes the sequence G arrow AATTC. So arrow will be the cleavage point. So whenever there is that site within the DNA, it will be cut. Okay. So you have many different type of enzyme. You have E. coli, you will cut at GAATTC. It comes from the Escherichia coli R bacteria. Hynate-3, you have the TTCGA and AGCTT for hemophilic influenzae. MHI, just the one that we just saw just now. GATCC, okay, bacillus amyloliquefaction. And TACI, a little bit shorter, TCGA, okay, from Thermus aquaticus. So, to make the recombinant DNA, you must treat both sources with the same excision endonuclease or excision enzyme. By MHI, for example, you cut the same site on both molecules, creating the GATCC sticky end. Okay, and this sticky end would have unpaired bases. So this unpaired bases can base pair with another DNA molecule that have the complementary sticky end. When they are bound together with live DNA ligase, they form hydrogen bonds with each other and they will be merged into one single DNA. Sticky end. Okay, if you can see there, one part has TCGC, and then the plasmid DNA, which are recombinant, also will have TCGC, but in another, in a backwards configuration. So because both of them match, then meaning that they can be joined together. Okay, so if you can see here, the gene within this round or ring-shaped DNA, which is called a plasmid. Okay, so let's say that each one of these parts of this ring-shaped DNA or plasmid create something. Okay, it is used for something, but since it is now being inserted with a new DNA, now the creation of the product cannot be done anymore, because the gene which is a white color is specified become useless as the new gene is added. So if you can see here, you have the DNA duplex. If it is cut with the enzyme, it will form sticky ends. In other cases, you also have the blunt end. So blunt end, you will make no overhanging bases. The base will be cut in a straight way. Okay. So if you can see SMA1, it cuts the base sequence of CCCGGG. So SMA1 cuts both DNA strands between C and G from a straight cut. So this DNA fragment does not have any blunt ends. Now I move to the next one, cloning vector. Cloning vectors, these are DNA molecules that are able to carry foreign DNA into host cell. So just like how mosquito is a vector for dengue, it just carries the disease. It does not get infected. Okay, just like that, the vectors for cloning also work the same way. It carries the foreign DNA into the host cell. It doesn't get infected by that DNA that they are carrying. Okay. For example, you have bacterial plasmids, bacteriophages, cosmids, and YACs. First of all is plasmids. These are the molecules of DNA which is ring-shaped in bacteria. It is separate from the bacterial chromosome. So it's very small, having a few thousand base pairs. Usually carry only one or a few genes and circular. They have only one single origin of replication, and they can replicate independently, freely and rapidly from bacterial chromosome. Meaning that during the cell division of bacteria, they do the binary fission. The ring-shaped chromosome, which is the plasmid, does not divide. Okay, the chromosome will divide, but the plasmid will not divide. It will just stay like that. So they are independent. Okay. Okay. So the vectors, the plasmid here, must be able to accept foreign DNA in multiple cloning sites. Okay, because many of these cloning sites, okay, would have many different enzymes for many enzymes. Okay, so it means that there are many places where the new DNA can be inserted. Okay. And this plasmid must have very useful gene, for example, AMPR, which codes for antibiotic resistance. So we use it to filter out which are the host cell that carry or do not carry the DNA that is of interest. We call it the genetic marker. Okay, the next one will be bacteriophages. So these are actually viruses, but they only infect bacteria. For example, Lambda 2001. So within this bacteriophage, they actually have a linear double helix DNA molecule. Okay, in recombinant DNA technology, a part of the bacteriophage DNA not required by replication is removed and replaced with the target gene for cloning. And then you put that DNA back into the virus, okay, and then this virus will infect the host cell with the recombinant DNA. Others, yeah, also have the cosmid, which is the SCOS1 combination between bacteriophage and plasmid. Okay, and then lastly, the YACs, yeast epithelial chromosomes. These are eukaryotic chromosomes and foreign DNAs. So bacterioplasmid, it is just for small DNA fragments, less than 10 kilobases long. Larger ones will be carried by the YACs. In order to replicate, the cloning vectors carrying target gene must be introduced into the host cell. The host cell, they must be able to accept foreign DNA and vectors and allow the vectors to multiply. For example, the most commonly used would be the E. coli bacteria, and sometimes eukaryotic cell can be used, such as the yeast or animal cell. So host cell must also accept the recombinant DNA through transformation, maintain structure of recombinant DNA from one generation to another, and also amplify the gene products of recombinant DNA. So we also need some enzymes to modify this DNA that we are using. For example, DNA ligase. So DNA ligase, they join fragments of DNA, catalyzes formation of phosphate ester bond with the sugar phosphate molecules of adjacent nucleotides. So meaning that they actually join the DNA fragments which are broken or unchained. So DNA ligase will chain them together. Okay, so we use it to combine the target gene and also the plasmid with the recombinant DNA. Okay, old and the new. Okay, that should be the tag polymerase. Tag polymerase would replace the DNA polymerase in PCR. Okay, because in PCR it's a little bit different. PCR we don't use any host cell at all. We just put the DNA in a test tube, and tag polymerase will do the rest. So tag polymerase will amplify short segments of DNA. So what does it mean by cloning? In cloning, we are creating many identical copies of the same recombinant molecule. So sometimes when we want to do analysis, we need more samples. Okay, instead of taking more samples from the host, why don't we clone that recombinant molecule many times to form material for analysis? Okay, so the thing is, cloning is referring to the cell organism derived from the parental organism. Okay, we use it as a term where we have the molecule recombinant DNA, which is original versus the copies of the original. So in animal cloning, if you can see here, you have organisms that are to be cloned, developing differentiated cells, and then replace egg which have been anucleated, which is have the nucleus removed, with the nucleus of the organism that we want to clone, and hopefully the nucleus will be developing into an embryo and creating a clone. Okay, so in gene cloning, what you need to do is, first of all, isolate target gene, insert it into vector, uh, transmit the vector into a host called transformation, and amplify target gene by the host cell by cloning and screening. So for cloning, what you have to do the first time will be what kind of gene that you want to get as the source. For example, if you wanna make more insulin, you take insulin gene from the pancreas. If you wanna make more, for example, saliva. Okay, take some saliva cells. Okay, so first of all, what will be done will be cutting the gene from complete chromosome using restriction enzymes and producing a complementary DNA. Okay, so there are two ways to isolate target gene. First of all is extract human DNA from donor cell, culture in lab, mix with restriction enzyme, separate the restriction enzyme using gel electrophoresis. Okay, electrophoresis will make the DNA to be sorted through the sizes. Okay, from light to heavy. A DNA probe is used to locate the target gene. Second method, using the cDNA. Okay, cDNA is the complementary DNA that we reverse transcribe an mRNA. So instead of getting the DNA itself, because DNA itself is like a very it's like a very complicated noodle or yarn. You cannot actually easily find that sequence that you want. So instead, you actually take the mRNA created, okay, mRNA created for that certain substance. For example, you have insulin. You look for the mRNA that created the insulin. Take that and use that as a template to synthesize the DNA. Usually, mRNA are made from DNA, but here, when you use reverse transcriptase, we use mRNA as a template to remake the DNA. Okay. If you can see here, this is how reverse transcription works. Okay. We have the DNA itself with intron and exon. Okay, intron, exon, you have the useful parts of the DNA, the useless part of DNA. Okay, and then you have RNA, okay, mRNA with the message that's carried by the DNA for transcription. Right, in transcription, the mRNA is supposed to be sent to the ribosome. Ribosome will create whatever the mRNA says. It's like a message. Okay, but instead, we actually capture the mRNA. We use the mRNA to rebuild the original DNA. Okay, so this is what you call the reverse transcription. So for mRNA, we reacquire the DNA, okay, using cDNA and DNA polymerase. Then we get the target gene. So when you get target gene, you can insert it into clonal vector like plasmid, bacteriophage, forming recombinant DNA. Usually bacterioplasmid would have marker gene to make sure that you are selecting the correct host cell that actually has that modification or recombinant. For example, you have MR and LAX-Z. So what is AMPR and LAX-Z? AMPR is the gene that makes them resistant. If you look at the name AMP, that's the short form for ampicillin. Ampicillin is the antibiotic. So if they had the MPR gene, they are resistant to the antibiotic. And then second will be LAX-Z gene. LAX-Z gene will be the code for synthesis of beta-galactosidase. Okay, this is an enzyme that can hydrolyze lactose and excal. So you can see here, you have the foreign DNA broken down with the restriction enzyme with the fragments and join them with the cut plasmid. Okay, and the parts which are overhanging will be joined, and in the end you have the recombinant DNA. As you can see here, this is actually a plasmid, right? Cut with BAM-HI. If you look at that yellow part right there, the yellow part is the AMPR or ampicillin resistant gene. Okay, and so when you insert that new gene of target in the target gene, for example the KN1, okay, blue one, when it's injected, it must be in the area which are not affecting the AMPR. Okay. Okay, so like this gene sequence contain restriction site for a specific restriction enzyme. If you can see there, right? That's the red color in that ring there. That's the LAC-Z. If this target gene inserted into that area, the LAC-Z gene will not function. Okay. So bacterial script from bacterial cell must be mixed with the same restriction enzyme cut the DNA molecule during the association of target gene. Okay, so when you mix them together, okay, we want to make them produce same sequence of sticky ends. So the hypothesis based on sticky ends will attract complementary bases of other sticky ends to form H bonds, what we call annealing. Okay, this annealing will be done in the temperature of 54 Celsius. Kind of like a little bit warm. Okay. DNA fragments are joined by the enzyme DNA ligase, which form covalent bonds between nucleotides and to produce recombinant plasmid. Okay. So what happens is that DNA ligase will glue them both together. So now you have two type of plasmids. One is a plasmid that do not carry target gene, because they kind of like repair their own plasmid, okay, re-annealing of their sticky ends. But the second one will be the one with the target gene. Why? Because they actually combine their plasmid with the target gene. Okay, so the one that carry, the one that don't carry. The newly formed recombinant DNA, which are plasmid carrying the target gene, can be inserted into appropriate hosts, most likely E. coli by process of transformation. So the thing is, plasmids are a ring-shaped DNA. Okay, how can we put them into the bacterial cell? Okay. So we induce the calcium chloride. Okay, we add calcium chloride in with the bacteria host cell, so that you induce permeability of the bacterial cell walls. So when that happens, the porous walls will allow the plasmid to enter the cells. Okay, so in the end, only a small amount of bacteria will take out the plasmid for the medium, and even smaller amount will take the plasmids which are recombinant. So what will be the three types of bacteria: one that do not have any plasmid, one that have non-recombinant plasmid, and the one that actually have the recombinant plasmid. So no plasmid at all, plasmid but no recombinant, plasmid with recombinant. So there are three types. So bacteria E. coli are cultured in a medium containing ampicillin and sugar, which is called X-gal. So this is where we will select which one carries the DNA of target. Okay. So the E. coli which has taken plasmid will grow and form colonies on the medium. Why? Because they have this gene called AMPR, ampicillin resistance. Okay, ampicillin resistance means that they can resist the antibiotic. Okay, because the medium of the growth for the bacteria actually has that antibiotic, the one that do not have the gene will die. The one that have the gene will live. Okay. Other than that, the bacteria that does not carry plasmid with intact like Z genes, they will produce. They are okay. For like Z gene, okay, if they have it, they can produce like Z and enzyme beta galactosidase that can hydrolyze S-gal. This will produce compound that makes colony blue color. But if the bacteria carries the like Z, which are not intact because it has been taken over by the target gene, then they will not make the colony blue. The colony will be, they will not make the colony blue. The colony will be what color? It will be white color. Okay, because like Z gene is supposed to be used to, hydrolyze X-gal to make it blue, right? But since the area where like Z genes used to be there is not taken over by the target gene, now it is non-functional. So it makes it easier for us to select which are the host cells that we want to multiply. We just select the white ones, not the blue ones, because white ones, they carry the recombinant gene, and also they are able to take in plasmids. So when we take them, they can divide very fast to produce new intelligent bacterial cells, and they can also express the gene to produce whatever they want to produce, for example, insulin. Here is the picture. Stage 1 is taking the bacterial cell plasmid and taking the target gene and treating both with the restriction enzyme to make sticky ends. Okay. And then when we add a DNA ligase, we join them together, and then we put them into the host cell. Some of the host cell actually have target gene. Okay, they are the one that actually have captured the plasmid with the target gene. Okay, and then they'll create two types of cells. Okay, the cells that form a colony of white and colony of blue. Colony of blue, they don't carry target gene because why? They still have the enzyme to hydrolyze S-gal. But the white color, okay, the white color, they are like that because they are unable to hydrolyze the S-gal sugar because the gene that is of target has taken over the place of LAC-Z. So we just select the white ones. So PCR. So PCR is something like the previous one, recombinants, but it doesn't actually need any host cell. It's like magic. Okay, it can be done in vitro, just in a test tube. Okay, so it can be done in if you look at in vitro, it is test tube. In vivo, it is what? It is in a living thing. For example, in microbe like E. coli, like yeast, which is eukaryotes, or in mammalian cells grown in tissue culture. For polymerase chain reaction or PCR, it is used to amplify DNA. It will produce many copies of specific fragments of DNA in vitro, which is in test tube. Okay, what you need is thermocycler, meaning that this machine can go hot and then cold and hot and cold again. Test tube containing the DNA sample, enzyme DNA polymerase, primers, and free DNA nucleotides. So if you can see here, cycle number one will create two molecules because the DNA will be broken into two due to heat. And because there's a lot of free DNA molecules in the test tube, together with the DNA polymerase, the polymerase will try to fix the DNA with the free nucleotides to make it whole again. Okay, from the half strand to become a full strand, and then in the end, because there are two half strands, these two halves will become two full strand of DNA. So from one, now you get two DNA. Okay, and then the process is repeated in cycle 2. Okay, in cycle 2, it heated up again, all of the DNA will be split in half again, and the DNA polymerase will do their job again, and from 4 halves it becomes 4 full DNA. After cycle 3, you get 8. Okay, so every time, every cycle, they will double and double and double and double. So after the 35th cycle, you will have 2 to the power of 36, which is 68 billion copies of the same DNA. So with all of the DNA, what can we do with it? We can make a DNA library. So DNA library would collect all of the clone DNA fragments. There are two types. One is the genomic library, which is the DNA fragments containing the whole genome of an entire organism, or the cDNA, which is complementary DNA. These are only complementary DNA molecules which are synthesized from mRNA molecules. Okay? They represent only part of cell's genome. So anytime a cell wants to do something, it will create the mRNA, and complementary DNA will be made out of this mRNA. So it is only capturing the DNA which are used for something. So genomic DNA is identical in all somatic cells. cDNA is just for the coding regions. So it is just complementary to cellular mRNA. So genome, okay, it is in every living thing. It is a complete set of chromosomes in a particular species. Each species has their own genome, different from the other organisms. The genomic library, it is a genomic DNA library that has all DNA sequences of a species. Once you have the library, you can be screened for clone containing sequence of interest. So why do we have this genomic library? Is to investigate the entire structure of gene and study regulation of the gene expression. So genome library would represent the entire genome from A to Z. It can be made in various vectors, but to keep that whole DNA of a certain animal or certain organism, we need a host to keep them. Okay, so to construct the genome library, total DNA extracted from somatic cell we cut with RE or gene enzymes. Once they are cut, we put it into each vector molecule, and what happens is that we will have a lot of compartments carrying a tiny bit of DNA. If you combine them all together, then you have the whole thing. Okay, just like when you watch a movie, sometimes you have this one, this two, right? The movie is like 2 hours long, but it has two VCDs. Okay, this one, this two. If it's just disc one, it will not be complete. You just can see the half of the movie. If you want to see the whole movie, you have to get two. So imagine your DNA is actually like that, but instead you have like 1,000 CDs. Okay, because why? Because we have a very big information in our DNA. It cannot be contained with just one single compartment or one single host cell. Okay, it must be put in a set of bacteria. Each of these bacteria will carry a small fragment of human DNA. You know, like this one, this two, this three, four, until this 1,000. Now we have the whole human DNA library. So for the cDNA library, it is a collection of bacterial clones that have recombinant plasmid with cDNA fragments. It is synthesized from mRNA with help of reverse transcriptase and contains only genes that are expressed in the transcription which are intron-free, meaning that you have the target gene which are useful and none of the filler stuff. Okay? Advantages: easier to control, as is smaller than easier to manipulate. But the bad thing of it is that it does not have the entire information of a certain organism. It is just the part of it that are expressed. There are many parts of our DNA which are not expressed at all, and they might be lost if we only use the cDNA library. Gene bank. It is a resource center for keeping and distributing genetic material and information for scientific community. So it is where we can actually, you know, if you want to have a reference of, you know, you want to compare our finding with a template, or if something that is standard, you go to a gene bank. Okay, let's say that you find a new species, you don't know what it is, you bring it to the gene bank and compare, okay, if it is matching or not. Okay, so gene bank is also stored in the form of gene library. Okay. So in the gene library, you have the seed, sperm, embryo that contain genetic material of species. So they are usually put in like cold rooms so that they will not degrade. A lot of developed countries have this, like West Germany and USA. They keep and distribute genetic material pertaining to humans, bacteria, viruses, agricultural plant, laboratory animals, and domestic animals. So in case that some animal became extinct, we still have their genes inside of the gene bank. Okay, application. Application of recombinant DNA technology can be done in agriculture, medicine, pharmaceutical, forensic, and basic research. So in agriculture, what's very popular is transgenic plants. So these are plants which are genetically modified to produce specific desirable and heritable traits. They are plants which are genetically modified to produce specific desirable and heritable traits. So these plants can be easily grown from a single cell using tissue culture method. So in this case, you don't need to plant a seed. You use a tissue culture, a class of cells, they become a new plant. For example, transgenic soybean. These soybean, they are resistant to glyphosate herbicides. So this herbicide will inhibit the enzyme EPSP synthase, which synthesizes essential amino acids that is required for growth in plants. Okay, and so plasmid can be used to insert extra copies of EPSP synthesis genes into crop plants that makes them resistant to glyphosate. Why is it important? Because when you want to spray the whole farm with a certain toxin, you want a toxin to kill the weed, not your plants. Okay, and so with the EPSP gene, they can resist against the pesticide. So example number two, maize. You know, corn. It is inserted with the gene for bacterium Bacillus thuringiensis that codes for production of protein which is toxic to insect paste of maize. So Bacillus thuringiensis is also called BT. So when the BT gene is put into maize, the maize will create the pesticide by itself. You don't need to spray. It creates a pesticide. Pesticide will kill the corn borer caterpillar. Okay. So when they try to eat the corn, they will be poisoned. They will die. Okay. Developing golden rice. So what is golden rice? It is a rice which are fortified with beta carotene, simulates the production of vitamin A. So widely used cloning vector will be the TR plasmid carried by Agrobacterium tumefaciens. So if you can see Agrobacterium tumefaciens, it is you get the plasmid, okay, and the plasmid we insert with DNA of interest to become recombinant, and this is the tDNA carrying the new gene will be regenerated a plant. The plant would have a new trait that we have injected. Transgenic animals. These are animals, they are improved by recombinant DNA. Okay, for example, the transgenic Atlantic salmon. So Atlantic salmon usually they grow very, very slowly. We inject them a new gene, okay, that will cause them to grow faster and reduce the time required to raise it to market size. So you don't have to actually wait for the whole year. You wait only half a year for them to grow up at full size. So the modified growth hormone genes, where they are inserted into the fertilized salmon eggs. Other than that, you also have the transgenic ruminant animals. Ruminants are animals such as sheep and goats, cows. Okay, they have their ruminants within their bellies. Okay, we inject to them a recombinant gene that causes them to create uh, pharmaceutical or therapeutic chemicals within their milk. For example, some people who have mutation of alpha-1 antitrypsin, they get this disease called emphysema or breathing difficulty. So where do you want to get this cure? Is by drinking the milk of the goats or cows that has the transgenic gene within them. Okay, so that's a way to actually get AAT protein is by drinking the transgenic animal's milk. So the thing is, this protein, it is not created in the animal, that create the milk, sheep and goats. We as humans, we put the gene into them, they will create it and they will pour it into the milk. Okay, and then what will happen? The baby will be a little bit cured because of the milk with this AT protein. You can see there. What is the origin of this gene? It is human. Okay, you put this human gene, Marco inject it into the egg of the goat and make it develop within a host. Now it grows up into a transgenic goat, a goat with a little bit of human DNA. So this DNA, okay, will produce a result. It will produce uh, milk, right? A milk that has the enzyme that humans need, okay, to cure that disease. Other products created in the milk of farm animals. You have the tissue plasminogen activator, TPA. This one is for treatment of heart attacks to dissolve blood clots. And second will be human growth hormone. So one of the most important usage of recombinants, production of insulin. Okay, why? Because before this, insulin was taken from animals. Okay, when you use animals, some animals they are forbidden. For example, in Islam, you cannot take anything that is made out of porcine origin, which is from pigs. Okay. Other than that, a lot of people actually get allergy due to the protein of the animals within the injection. Okay, and sometimes the disease from the animal can jump into the human when they are injected with the insulin from the animals. So instead, now we have the insulin created by a bacteria called E. coli. So E. coli does not naturally create insulin. Humans create insulin. Mammals create insulin. Okay. So how did we do it? Okay. So for example, if you are diabetic, you need insulin. Okay, and the insulin will be in a small bottle that you can inject. But who created it? Okay, it is the bacteria that creates it, but with the help of a human gene. So the first thing first, the gene for insulin must be taken. Where? From pancreas, because pancreas creates insulin. Take that gene, put it into a plasmid, and put it into a bacterium. Okay. So this bacterium now has the ability to create insulin. Even though they are not born with that ability, they now have the ability. So when they multiply, they can also create even more and more and more insulin in the fermentation tank. So what we do as a human is just harvest them. Okay? In this creation of insulin by bacteria, there's no involvement of any animals at all, just human and bacteria. So another usage would be in the environment, oil spills. Okay, we put the gene for digesting hydrocarbon into the bacteria, and we just we just release the bacteria into the oil spill. The bacteria will eat the oil, right? For example, it's called Pseudomonas. Pseudomonas, they are genetically altered to break down hydrocarbon in oil to clean up the oil spills. So we call it the bioremediation. Okay, we use biology to clean the pollution. So DNA diagnosis in medicine, we have the big project called the Human Genome Project. So it is a project that aims to map the location of all human genes in all chromosomes of humans. So in human genome, we have more than 1,000 genes, and there's 50 alleles per chromosome. So mean that the human DNA is actually very, very vast. Okay, so genetic map of chromosome can help tailor treatments to an individual. Okay, mean that if you can see what kind of disease that is in that person, you can make a treatment just for the person. Okay, for example, you know what is hypertension? You know, you have the high blood pressure, right? So people used to say, okay, you have high blood pressure, eat less salt. But the thing is, eating less salt only works in just a small percentage of people with hypertension. Some people with hypertension, even though they eat less salt, they still have hypertension. It is caused by another cause. Okay. Um, so current tests for mutant angiotensinogen may correlate with benefiting from a low salt diet. So if you actually have a mutant angiotensinogen, which is a protein that gives a signal for the kidney to, absorb salt reclamation, then it might cause your blood to have high blood pressure. So those type of people might be able to benefit from the low salt diet, but others probably cannot. So mutant genes have been correlated with the specific drug treatment. For example, medicine can carry label as effective only to a certain people. For example, people who are treated by the new Glaxo Wellcome drug can only treat somebody with the mutant gene for apolipoprotein E. This is a substance that is only created by the brain of the person with Alzheimer's. So other than that, there's also this mutant gene for Sarcopenia. This is just for cancer. It is treated by ammonophyte. Okay. Other than that, the mutant gene for chloride gate, which is for the disease cirrhosis, is treated by pomozine. And another one would be mutant gene for angiotensinogen, which is hypertension, treated by a low salt diet. Okay. So all of the diseases only are effective for a person with a certain genotype. So how do you do with genetic screening? Okay. It is by prenatal, carrier, or predictive. Okay. So genetic screening can actually help us find a disease very, very early in life. For prenatal diagnosis, natal means born. It is before somebody is born, we actually screen them for any diseases. So if the disease has been identified, we will tell the parent, okay, give counseling, okay, what is the expected lifespan, what are the quality of life, what the baby might suffer when they are born, and the parent will be given an option for abortion if the baby really cannot survive with these genes that is ailing them. Other than that, there's also carrier diagnosis. Diagnose somebody's DNA, look whether they are carrier of a disease. Okay, some people they look healthy, right? They don't have any diseases, but they carry the gene for the diseases. So they might not have the disease, the children will have the disease. Okay, so if a couple has been screened to have this carrier of a certain gene, then um, if they want to have children, we or doctors can discuss with the parent what would happen if they have children. What are the chances to get the disease baby and other issues involved. And the last one, predictive diagnosis. If you look at the name, okay, predictive means that you are using it to predict something. Okay, so they can show potential of developing some diseases as a result of presence of some particular genes, but doesn't happen yet. So some diseases, you actually have the gene, but the disease doesn't happen ASAP. It happens later in life. For example, Huntington's chorea. Okay, in that you are unable to move your body because of Huntington disease, because your brain becomes shrunken. This gene only happens, this gene when it's detected, it only manifests when somebody is in middle age. When somebody is in their 30s, then they will start to manifest the disease. So some of the important techniques used in this diagnosis will be chorionic vector sampling, means that you are actually taking some samples from the baby's, the placenta, amniocentesis, which is the water within the womb, and gene probes. So you can see here, this is how the um, genetic screening would take the sample. Okay, under guidance of ultrasound, sterile needle is inserted into abdominal wall into amniotic sac. Okay, and then amniotic fluid is taken, withdrawn to the needle. And then what will happen is that in this liquid, okay, surrounding the baby, there might be the cells from the baby itself, the fetal cells. We culture the cells and we test the cells. Okay, chemical analysis, DNA analysis, and chromosomal analysis. So once we know that somebody has a genetic disease, there might be a chance for gene therapy. Okay, some people are born with bad genes. Okay, they cannot escape the disease that is being inherited. So we give them gene therapy to make up for the faulty gene. Okay, we use it to treat genetic disorders and various human illnesses. The two methods, ex vivo and in vivo. Okay. So ex vivo, we insert the gene into the individual cell to treat a disease and hereditary diseases. Okay. So you have the mutant gene in your body that causes the disease. We want to replace that gene with a correct gene, okay, using a retrovirus. For example, children with an SCID syndrome. What does it mean? It is a severe combined immunodeficiency syndrome. These children do not have any immune system at all. Okay, so why? Because they do not have the enzyme adenosine deaminase (ADA). This is a very important enzyme that causes the T and B cell to be mature. So these T and B cells, they are the one that attacks or defends your body from any pathogen. If they don't have this, the whole life will be unprotected. Okay, so how does gene therapy help? Remove all of the bone marrow stem cells and infect them with the retrovirus that carry a normal gene. So this retrovirus will infect the stem cells with the correct gene to replace the faulty gene. So use of bone marrow stem cells will allow them to divide and produce more cells with the same genes. Why? Because stem cells in the bone marrow is where your new cells are created. Okay, all of your cells come from the bone marrow stem cell. Bone marrow stem cell will divide every day to produce new cells for your body. So when we treat the disease, treat the gene therapy from the source, meaning that all of the new cells which are being born, they will be created with the correct genes. Okay. Okay. You can see here, you have the bacterium claudine plasmid with normal human ADA gene with a genetically disabled retrovirus. So the retrovirus will be the vector. Put that gene into the virus. The virus will infect the bone marrow cell, and then what will happen is that they will transfer that gene into the cells. So now the cells actually have the correct gene. So we grow them in culture to ensure that ADA gene is active, and we even reimplant the cell with the correct gene into the baby. So now the baby can produce the enzyme called ADA. Other gene therapy trials, you have the familial hypercholesterolemia. Okay, if you look at the name, familial means that it runs in a family. Hyper means high. Cholesterol, it is cholesterol. Mia means blood. So people who have this gene, the whole family have high blood pressure. Why? Because so much cholesterol in the blood. Why? Because they have liver cells lack a receptor for removing cholesterol from blood. Means that they naturally have high cholesterol. Okay. Next will be cystic fibrosis. Cystic fibrosis will be a disease where your mucus becomes so thick like glue, it is really hard to breathe at all because there's no gene for transmembrane chloride ion carriers. So next will be cancer treatments. The bone marrow stem cell from a woman with ovarian cancer will be infected with a virus carrying gene. made them more tolerant to chemotherapy and cancer cells much more vulnerable to chemotherapy. So, I mean that chemotherapy is a treatment that causes all dividing cells to slowly suffer and die. Okay, but with this treatment using gene therapy, the good cells that we don't want them to die, they will survive, but the cancer cells will die quickly through the chemotherapy. DNA fingerprinting, we use it for identifying criminals, dead victims beyond recognition, like a burn victim. Okay, when you have a burn victim or somebody who has been dead for a long time, or somebody who has been flattened, okay, you cannot use their fingerprints anymore. Okay, you take their DNA and use it to identify the person. Other than that, it is used for paternity cases, okay, to determine is this child, you know, is the child of this parent or not. Okay. So a procedure called RFLP analysis is involved, which is the residual fragment length polymorphism. So 90% of human DNA do not code for proteins and has no function at all. So the base sequences from the non-functional segments. We have 20 to 40 bases. We use it for DNA fingerprints. So you have the blood sample, collect the blood sample, get the DNA, okay, separate the fragments through electrophoresis, and you form a band pattern on the gel, transfer into null membrane, and then DNA probe will bind to specific DNA sequences on a membrane, and as the DNA probe washed off, at this stage you have the radioactivity probe bound to DNA pattern, and you shoot it with the X-ray, okay, with the X-ray film, then the film will be developed. What you can see are bands, thick and thin bands. These are the DNA fingerprints. Okay. The thing is, DNA fingerprint, it is very specific to a person, just like our fingerprint on our finger. Okay, every person will have a specific DNA fingerprint. So in criminal cases, it is used to establish a link between biological evidence and suspect of a criminal investigation. So if their DNA is found, okay, in the crime scene, it will match the suspect. Okay, if two of them match, then the evidence comes from the suspect. If they do not match, then evidence say that he was not there. It doesn't come from the suspect. So you can see, if you collect the DNA from crime scene, and then suspect A, suspect B, if they match, they'll have a band which are similar. If you look at this picture, suspect A, suspect B, okay, suspect A has one band, suspect B has two, and the two band are similar with the crime scene evidence. So suspect B is the killer. So this is the application of DNA fingerprinting in parentage. If you can see here, mother and child and alleged father. Okay, if they are the correct parents, they would have the same bands of the DNA. Okay, usually for mother and child, there will be an area which are the same. We can see uh, there are parts which are lining up. Okay, if they are not the actual father, then they will not have any matches at all. Okay. So this is a recap. These are references. Thank you, and I'll see you in the next chapter.